this. It is the EBS PhD contest, where we'll be hearing from 3 early career researchers who will be presenting their research. Just before we get started, a quick thank you to our judges for this session, who will be judging the best talk. Julie Cook from the Monash Department of Econometrics Business Statistics, Mahsa Salehi from the Faculty of IT at Monash University, and Lauren Smith from the Walter and Eliza Hall Institute in Melbourne. Okay, let's get started with our first speaker, who is Malvika Kharbanda. Malvika, do you want to share your screen, and I'll just read out a brief introduction?
Yes, let me share my screen.
Great. Malvika is a PhD candidate from the South Australian immuniGEnomics Cancer Institute. She's also the current treasurer of Combine, which is the student subcommittee of the Australian Computational Biology and Bioinformatics Society. Today, Malvika will be presenting to us on evaluating the prognostic potential of transcription factor activity in prostate cancer. Thanks, take it away.
Thank you, Michael. I would like to thank all of you for giving me the opportunity to present today. And I'm speaking from the land of the Ghana people, and I'll be taking you through my PhD project, which is evaluating the prognostic potential of transcription factor activity in prostate cancers. Now, when we think about cancer, one of the first questions is that, can we predict which patients are at risk of cancers? And the answer to that is yes. This is the power of modern data science in medicine, which allows us to predict what the disease outcome will be by quantifying molecular data. And today I will talk you through how we can use large-scale molecular data to make these predictions. Now, to understand how data science can unravel the mysteries of cancer, let's start with the basics, which is the central dogma of biology. Now, the central dogma of biology dictates that the DNA in every cell holds the genetic instructions to make the proteins that are needed for life. The process starts with transcription, where the DNA is converted into the RNA, and then the RNA is used to create the proteins. Now, the transcription factor here is responsible for what genes get transcribed from the DNA to the mRNA. And we can think of the transcription factor like a head chef at a restaurant who makes the dish according to what the customers tell him or tell them to do. And why is this important in cancers? Is that in cancers, certain genes that regulate cell growth or division can become dysregulated. Now, the transcription factors that control these genes often become overactive, leading to uncontrolled cell growth, which is a hallmark of cancer. So an overexpression of transcription factor can lead to an overexpression of cells, which is in fact the tumor, and it spoils the dish. So this is where our work begins, by studying the changes in gene expression, which can identify the key players that drive cancers, and can also predict how aggressive the disease might become. Now, prostate cancer is a global health challenge. Why is it a global health challenge? Because 1 in 7 men are diagnosed with prostate cancer globally, making it the second most common cancers in men worldwide. So the prostate is a small walnut-sized gland that is important for reproduction in men. And this also becomes a source of cancer as men age. Now, small changes in the DNA can cause the cells to multiply faster and live longer than they normally would. When the cancer is localized in just the prostate tissue, that's called the localized tumor. But when the cancer progresses and moves to other parts of the body, that's called the metastatic cancer. And most men in prostate cancers cases are diagnosed at the localized stage, where the standard of treatment is a radical prostatectomy or a surgical removal of the prostate. However, around 30% of the men move on to becoming a metastatic tumor case. And this is where molecular data comes in, so that we can identify, by looking deeper into the gene activity, which tumors become metastatic and which do not. So how do we capture this gene activity to identify which genes are overexpressed on which become cancer? In early 2000s, a breakthrough technology called the microarray transformed the biology into a data-driven science. Now, this allowed us to measure the expression of thousands of genes at once. Now, the microarray works as a technique where thousands of DNA probes are fixed onto a chip. And when we extract the RNA from a tumor sample and apply it to the chip, the RNA then binds to the complementary DNA that's present on the chip. And it gives us a signal which tells us eventually what genes are expressed in what samples. So it gives us a gene expression matrix at the end where we have each sample and the value of the gene expressed in each of the samples. By looking at this snapshot of gene expression profiles from the microarray, we can then create a snapshot of all the active genes in the cancer or normal cells. And these profiles give us clues on what might be happening inside the tumor and the genes that drive the cell growth. Now, coming back to prostate cancer. So prostate cancer is a hormone-dependent cancer. Therefore, when you look at metastatic prostate cancer cases, the standard of care is to treat the patients with androgen deprivation therapy or ADT. And now this charter trial was pivotal in studying the difference in treatment arms for metastatic prostate cancer patients. And this was led by my principal supervisor, Professor Christopher Sweeney. Now, the charter trial is a phase 3 clinical trial, which is a microarray data set of 159 patients diagnosed with metastatic prostate cancers. And they were randomly treated with androgen deprivation therapy and androgen deprivation therapy plus docetaxel. Now, docetaxel is a chemotherapy drug. And one of the key findings of the chartered clinical trial was that adding docetaxel to androgen deprivation therapy significantly improved overall survival in patients with high volume disease. Now, some patients experienced long-lasting benefits, while others progressed more rapidly despite this aggressive treatment. The other molecular insight that we got from the chartered trial was the identification of this gene called the MIBL2. Now, MIBL2 is a transcription factor whose high expression was linked to poor survival despite aggressive therapy. Now, identification of a molecular marker like MIBL2 can help us predict which patients are at a higher risk of progression and which may benefit from additional or alternate therapies. MIBL2 plays a critical role in regulating genes that control cell division in cell division. But in normal cells, MIBL2 is tightly controlled, whereas in cancer, its overexpression can drive uncontrolled growth. MIBL2 controls downstream genes as well that promote tumor progression. So MIBL2 essentially is a transcription factor that controls the activation or suppression of other genes downstream of it. Now, in a completely independent study by the people here, including Dr. Natalie Lister, who is my co-supervisor, they identified MIBL2 as a master regulator of prostate cancer. They used the Mogrify gene prediction algorithm in a group of 42 PDXs, where MIBL2 was one of the highly ranked transcription factors, and that drove aggressive prostate cancer disease. Now, the Mogrify algorithm also gave us a list of target genes that are controlled by MIBL2. So it gave us the transcription factors that are important in prostate cancer, and it also gave us the targets of all of these transcription factors. So along with MIBL2, there were other 42 transcription factors that were predicted by Mogrify. Now, what we had was these important transcription factors, and we had their networks. So we decided to score the network activity. And one way to score the network activity was to use this package in R called SyncScore, which was developed by my co-supervisor, Dr. Dharmesh Bhuva. And currently, I'm the maintainer of the package on Bioconductor as well. So we used the SyncScore algorithm to quantify the network activity of these transcription factors. How SyncScore does that is that it takes the expression data as input along with the network genes. So the expression data is just the expression of each gene in that particular sample. And then the algorithm ranks all of these genes in that particular sample. It takes the ranks of only those genes that are present in this network And it gives us a combined score, which is just the sum of the ranks of the genes present in the network divided by the number of genes in this network. So we have the expression of the transcription factor, and we have the transcription factor activity score. And what we wanted to do was to evaluate the prognostic potential of these transcription factors and transcription factor networks. So a prognostic biomarker as such is a gene that provides information about overall outcomes of a course of disease, regardless of the treatment. It indicates the likely progression of a disease, such as how aggressive it is or the patient's chance of survival. Now, to determine the prognostic potential of MIBL2, we use the Cox hazard proportional model. Cox hazard proportional model is a regression model that analyzes the relationship between time to an event, such as progression or death, and the other explanatory variables. And here the explanatory variables are either the gene expression or the network activity score. The Cox model can be thought of like a multiple linear regression where we are modeling the log of the hazards. And instead of predicting the raw survival times, the model predicts how the log hazards changes with different covariates. Now, simultaneously, it also evaluates the effect of multiple factors on survival. So multiple factors as in the age of the patient or the tumor volume of the patient. So the output that we get from the Cox hazard proportional model is a hazards ratio, which measures how each factor affects the risk. So if a hazard ratio is greater than 1, the factor increases the risk of the event happening there, such as death or progression. And if the hazard ratio is less than 1, it tells us that the factor decreases the risk of the event happening. And here the factor is the gene. So we fit the Cox hazard proportional model in the chartered trial, where we use the 42 transcription factors predicted by Mogrify, and we just used the expression values of the genes in the Cox model. And when we did that, we found that MIBL2 and the other important aggressive prostate cancer transcription factors were not prognostically significant in this data set, which got us to thinking that what if we look at the expression or the activity of the transcription factor or the downstream targets. So we used SynScore to score the activity of the transcription factor, and we found that yes, the MIBL2 network activity was highly prognostically relevant in the chartered trial or in aggressive prostate cancer patients. So it proved our hypothesis that transcription factor activity scores provide a stronger prognostic insight compared to transcription factor levels alone. And this can help us identify patients at risk of poor outcomes, allowing for a more personalized treatment approaches. Now, moving on from metastatic prostate cancers, we moved on to localized disease, where we have this entire cohort or an atlas of cancer called the Cancer Genome Atlas, where we have 33 different cancers and also the clinical metadata for localized disease. And we tested the prognostic potential of all the transcription factors and their activity in these 33 cancers. And this is the prostate adenocarcinoma data from the TCGA data sets. But one interesting thing we found from this is that the MIBL2 and the MIBL2 activity score showed high hazard ratios across several data sets, indicating that they're consistently associated with an increased risk or worse prognosis in various cancers. And these are mostly solid tumors. And now our lab is focusing on developing therapies that can directly inhibit MIBL2 and its downstream effects, offering a new approach to treat men with high risk or aggressive prostate cancers who do not respond well to the existing therapies, such as radical prostatectomy or androgen deprivation therapy with chemotherapy. And we can also use the therapy or the therapeutics that are developed to inhibit the MIBL2 expression for other cancers that are relevant, such as kidney chromophobe, where the hazard ratio is extremely high for the MIBL2 activity, proving that it might be extremely important in driving these cancers. Now, our future work is to extend the SING-SCORE work into a more spatial transcriptomics side as well. And in spatial transcriptomics, what happens is that there are the technology gives us a snapshot of all the genes present in a single cell at a spatial resolution. So it tells us if a cell is here, what transcription factors or what genes are being expressed there. But one limitation of spatial transcriptomics at the moment is that it has a panel of only 300 to 400 genes, whereas the whole transcriptomic microarray data gives us more than 18,000 genes in a sample. So here you can see this is an H&E image. So the H&E image is a stained biopsy that is usually looked by a pathologist to identify which areas are cancer and not cancer. This is what is done in clinics. So now we have that image. We also have the spatial transcriptomic data. So we have the transcription factors or the transcript information for each gene. And we can use SING-SCORE to score a group of genes which are also called the hallmark androgen response. So we have a group of genes that are known to have known to be high in prostate cancers. And we see that these regions are high in this hallmark androgen response, which are also the prostate cancer regions. And also the same regions are also high in a fatty acid metabolism pathway. So there is a hypothesis ongoing that a high fatty diet might lead to prostate cancers as well. So that's led by another supervisor of mine called Lisa Butler. I would like to thank my team and all the men who participated in the clinical trials, making my research possible. And I'm open to questions.
Thank you, Malvika. Very interesting. Do we have questions from the panel or the floor for Malvika? I have a question for you. On your chart about the hazard ratios, the heat map of the hazard ratios for the different cancers, I noticed that some of these are green and so less than 1.
Yep.
What does that mean in this setting?
So in that setting, it means that transcription factor is related to good survival. So that might be a gene that's good in that particular cancer, and that leads to your so that's a good gene basically, and you will not die as fast. Great.
It's not all red, so that's possibly a better outcome.
Yeah, so it's a better outcome. Yeah.
Other questions? Okay, if not, let's thank Malvika again. Our next presenter in this session is Felicia Bongiovanni. Felicia is a PhD candidate from the Walter and Eliza Hall Institute in Victoria, Australia. And she will be presenting her work titled Inferring Time of Infection by Understanding Antibody Dynamics Through a Hierarchical Bayesian Framework.
Thanks, Michael. I'll just quickly share my screen. Amazing. So thank you for the introduction, and thank you for everyone to attending this talk. Abracadabra. It's a phrase that we have most likely all heard at some point, and we normally associate it with magic tricks or movies. And although we don't know the origin of this word, we do know when it was first documented and when it first appeared, which was in the 2nd century AD by a physician to the Roman emperor. And abracadabra was actually used on an amulet. It was written on an amulet in a triangle form seen here and prescribed to sufferers of malaria. Was this an effective treatment? I will use it to your best judgment to decide that. But let's just say at some point in time, around that point in time, malaria was on every continent of the earth except Antarctica. Since then, we have significantly reduced the burden of malaria with effective drugs. However, it does still pose a significant public health concern, where nearly half of the world's population is still at risk in areas at risk of malaria transmission. In 2022 alone, there were 249 million cases and 608 deaths reported globally. There are several parasites that cause malaria in humans, the two main ones being Plasmodium falciparum, which causes the highest burden in Africa, and Plasmodium vivax, which is much more widespread and is predominantly in the Asia Pacific. One of the main challenges that come with eliminating Plasmodium vivax is the complexity of its life cycle. Now, all of the parasites that cause malaria in humans share a very similar life cycle. It starts with a mosquito taking a blood meal from a human. The parasite will enter the body and travel to the liver, where it will undergo some further development before it then starts infecting the red blood cells and going through asexual reproduction. After asexual reproduction, some of the parasites will take on sexual forms, which are taken up by a mosquito once another blood is taken. A unique stage in this life cycle where some of the parasites upon first infection will undergo the normal life cycle. However, some will remain dormant in the liver and they are clinically silent. So none of our current tools can detect the parasite while it's in this stage. And up to 80% of all blood stage infections for Plasmodium vivax are caused by these hypnozoites relapsing, which usually occurs months to years later. So in order to eliminate Plasmodium vivax and reduce the burden of it, we need to be treating people for these hypnozoites, but we can't directly detect them. So what does that leave us with? My lab at WeHi has developed a new strategy where we can maybe indirectly infer people that are likely carrying these hypnozoites through measuring their antibody levels. So the reasoning for it is that your first relapse after an infection usually occurs no more than 6 to 9 months after the mosquito bite. And we also know that that first blood stage infection that you have will induce antibody responses that are long-lived. So it gives us a new approach. We can measure antibody responses to a panel of Plasmodium vivax proteins that can act as serological exposure markers. So we can identify people that are either currently infected or have been recently infected in the last 9 months. And those will be our likely hypnozoite areas. So the challenge that we're trying to find a solution to, and what my PhD research is focused on, is the ultimate goal is to develop a tool where someone will provide a blood sample, a single blood sample from which we obtain a single antibody measurement, and we estimate how long has it been since they were infected by the mosquito and they had that primary blood stage infection. And this is possible. Once we understand the dynamics of the antibody response, we can translate this blood sample into a time of infection. But how do we understand the dynamics that are underpinning the antibody response? We can use mathematical models. So this is the antibody kinetics model that I'm using in my research, which describes the different increases and decreases in the antibody levels over time. So it accounts firstly for any pre-existing antibodies that you have from a previous infection and how they are decaying over time. And it accounts for boosting and decaying of antibodies secreted by short-lived plasma cells and long-lived plasma cells. So this is a biphasic decay where you have two different cells decaying at different rates and they're both secreting antibodies. And this is a lot more reflective of real-world biology of what's happening internally during an infection. So what do we actually do with this model? How does this help us understand the antibody response more? Well, all of these fancy letters in the model are called parameters, and these parameters are unknown values that once we know the values of them, we can be calculating antibody levels at a certain time. And these model predicted antibody levels, we want to be replicating real world data that we have. So we want to fit these parameters to observe data from the real world to understand the dynamics of the antibody response. Things like what is the half-life of an antibody? What is the half-life of your overall protection during infection? So the kind of data that I'm talking about that we need for this approach is antibody measurements for individuals over time against a panel of proteins for Plasmodium vivax. And what we do with this is we estimate all those parameters using Bayesian inference through a probabilistic programming language called STAN. And Bayesian inference is essentially built upon Bayes' theorem, where we are trying to calculate the probability that our parameters equal a certain value, given the data that we've collected from the real world, the data that we've measured in the lab. And that's proportional to the probability that we have our data given parameters equaling a certain value. So that's our likelihood times the probability of our parameters. So that is our prior beliefs on what these parameters should be. So we can have a very confident belief that, say, the half-life of an antibody is around 21 days, or we can have no idea where the value of a parameter will lie, and we can communicate that through something called a prior distribution. So all of these come together for us to understand the antibody dynamics more. And this approach that we're using is in a hierarchical sense. So what that means is that we have a population of individuals that we have data for. And for each of those parameters, we're trying to estimate what the distribution is at a population level and an individual level. So the purpose of this is that the population level distribution will cover all of the variation between the individuals in the population, and of course, the individual levels that we obtain the value for each specific individual. And these both get put into the model. So while we're fitting the parameters of the equation, we're putting into the model the individual level parameters. But when it comes to estimating the time of infection for that single individual that we have no information about, we'll be heavily relying on this population level parameter. So we know the individual belongs in the population. Here's the possible range of values that their parameters could be in order to estimate that time of infection. So putting all of that together, what have we done so far with this? So far, I've simulated the IgG response. So IgG is a specific class of antibody for two different populations, for 100 individuals only exposed once to an infection, and 100 individuals that were either infected once or multiple times. So we modeled the full antibody response to 99 of them to obtain those posterior population level parameter estimates. And then for a randomly selected individual, we randomly selected a single antibody measurement from their collective antibody measurements and estimated that time of infection. So what that looks like, here are two randomly selected individuals, and for each of them, we have two different selected antibody responses. So for this individual, we selected an antibody measurement just over 100 days from diagnosis, from exposure, sorry. And then for this individual, over 300 days. And we pretend that we don't know this. We don't know how long ago they were actually exposed. We do the approach, and these are the results that we get. So for the first individual, we get quite accurate and confident results that the individual was infected just over 125 days ago. And the same with the individual below, we get quite accurate and confident results shown by the red line being the true value, how long ago they were infected, and this black line being the possible values that we estimate to be their time of infection. And if we look at the actual statistics of this, we can see how close we get to estimating. So true exposure was 130 days ago. The mean posterior estimate for this was 132 days. And our mean absolute error was about 8 days. So we don't deviate much from the true value. The below individual was around 322 days for the true exposure, and our mean estimate was the same, and a mean absolute error of around 8 days as well. So this is great, but we want to make this a bit more of a real world example now. So what if we have individuals that have been infected multiple times and we have an antibody measurement after multiple exposures? So that's what we have here. This individual, we select their antibody measurement after their second exposure, and this individual quite a while after their second exposure. And in order to do this, we need to change our assumptions about any pre-existing antibody levels these people have. So if it's after a first exposure, we can be confident that they have no pre-existing antibodies. They've never seen this infection before. After a second exposure, they will have some pre-existing antibodies. So all we need to do is come back to the equation, and we need to change our assumption about this parameter here and re-estimate pre-existing antibodies. And after we do that and we perform the same approach, we get accurate results still, but there is a lot more variation in the estimates that we obtain, and it is less confident. So the true exposure for this individual was 90 days, and our mean estimate is 95 days, but the mean absolute error is around 45 days. So there's a much greater deviation from the true exposure in our estimates. And the same happens with this individual here, where their true exposure was 306 days ago, mean estimate of 354 days, and mean absolute error of 55 days. We were expecting that these results would have a lot more variance the longer we get away from exposure, because the antibody levels will plateau a lot more. But this does show how there's a lot more uncertainty after a lot more exposures. So in order to make this a more accurate approach and to improve it, there are some things we want to do. First, we want to reparameterize this model. So instead of estimating the time of exposure, we'll estimate the time since peak of infection. We'd also like to incorporate multiple antigens or multiple antibody classes to provide more information to the approach. And then we want to apply it to actual observed data. So this has been simulated data so far. Then we want to apply it to COVID profile data. So that's observed data where we have confirmed diagnosis dates for people. And then Plasmodium vivax cohort data will be real world data with no accurate confirmed diagnosis dates of people, and where we answer the question of inferring hypnozoites or hypnozoite carriers. So I'd like to take a quick moment to thank everyone that has contributed to this work so far, both on the modeling side and on the wet lab side. And I'm happy to take any questions.
Great. Thank you, Felicia. Do we have any questions from the floor? Felicia, I have a question for you. I noticed that all of your estimates in the second set of models, so when you were looking at the dual spike, all of them, the mean estimates were higher than the true. Is that true across all patients, or does that just happen to be for these ones? And do you think there's any reason why that might be?
So with the mean estimate, it does depend on the individual that we've selected, and it does depend on how far from the exposure date they are. The closer to the exposure date, we get more so like either just before or just after. And then the further away we're getting more after the fact because of one, the observed error, the observational error in the model, and also a lot more variance in, that antibody plateau. So it does kind of depend on the individual and their own parameter estimates, but I do see a pattern with that occurring further away from infection you get. And you can kind of see that with these two individuals here. This person, the estimates are, there is a long tail here, but the mode of it is kind of more just before, and this individual it's right after, and they're further away from exposure.
Great. And a question from the floor for you. What is the real world impact of that fairly large uncertainty or error in estimating this time of exposure?
So the real world impact would be, like I said in the beginning, the kind of cutoff for a likely hypnozoite carrier is 9 months. So if you're infected in the last 9 months, we'd identify you as a likely hypnozoite carrier. If there is a lot of uncertainty around whether someone was at the 9-month mark or maybe at 12 months or something like that, and we're in a setting that has limited resources, that person may get passed in terms of treatment, which is something we don't want. We want every individual carrying hypnozoites to get treated. It's the only way to reduce this burden of Plasmodium vivax. So that's why it's really important for us to get accurate estimates here so no one gets bypassed due to limited resources or anything else.
Great. Let's thank Felicia for that presentation.
Can I ask a
sorry,
question?
another question? Yes, of course.
Sure. Thank you so much for the great presentation. So in terms of this multiple exposure case, you said that it's a synthetic data, right? So can you explain
Yes.
how, so how did you, so is the exposure, like this multiple exposures, the same among the individuals? Is it just random? Could you explain a little bit more on that? So because I just wanted to understand the source of variation, why, the mean absolute error is, for example, much larger than the previous case. Just wanted to understand where this variation is coming from a bit more.
Yeah, of course. So based on the results I've seen across doing this across many people, even though I'm only showing 2, I have found that the mean absolute error gets much larger the further away the point of exposure is, not when, like how far the antibody measurement we've collected is from the time of exposure, not actually because the time of exposure was at 200 days as opposed to 100 days or anything like that. Because as we get further away from that time of exposure, the antibody levels are being secreted at a more constant rate and they're plateauing. So it kind of becomes a wider range of this antibody level we could be measuring either over here or way over here. And as for how we decided when these exposure times were, it's just randomly selected for each individual.
Okay, right. Thank you. Thanks so much.
Thanks.
Are there any other questions for Felicia? Okay, if not, let's thank Felicia for her presentation. And we will move on to our last presentation for this session, which is from Simisana Nbada. Simisana is an early career researcher from the Department of Computer Science at the University of Botswana. She's also a very active member of the Our Ladies Habaron. She'll be presenting today on a review of the use of R programming for data science research in Botswana.
Hello, everyone. Let me just share my screen. Can we all see my screen?
Yes,
Is that
looks
okay?
great.
Okay. Yes. Thank you, everyone, for sticking around to listen to my presentation. I'm Simisana Nbada. We'll be presenting a review of the use of R programming in data science research in Botswana. To go through the overview of the talk, I'll be going through the research background in Botswana, the objective of the study, the Prisma methodology that was used, and the R programming used in Botswana data science research, and the discussion as in the findings that were found. So where I'm from Botswana, which is a landlocked country in the southern part of Africa. More than 80% of the country is made up of the Kalahari Desert, and it's home to wildlife like elephants, giraffes, crocodiles, and lions. So research in Botswana is guided by the Monuments and Relics Act, which is the protection and safekeeping of ancient buildings in Botswana. While development-oriented research is a priority in the interest of expanding knowledge in various fields, research of a more academic and theoretical nature is permitted wherever possible. The research topics with the greatest number of publications are in the areas of geoscience, ecology, environmental science, water resources, and veterinary sciences. Botswana has a national research, science and technology plan, which is informed by the need for the Center of Excellence to earn a reputation as a significant resource for the progress of STEM studies and the spread of innovation. Exploratory and descriptive statistics in Botswana research is done with data, SPSS, and Atlas TI. R programming is mostly used by statisticians. Despite its growing use in data science, very few research conducted in Botswana have used R programming for their data analysis. So the objective of this review was to provide a snapshot of the data science research in Botswana that have used R programming for their data analysis experiments. Key reviews have demonstrated Python programming to be robust, scalable for data analysis, but no review has comprehensively mapped all program applications within the context of research in Botswana. So such a review would equip both data scientists and R practitioners in Botswana in the methods and applications of R programming. It would also give insight of the challenges of using R programming in Botswana, as well as identifying gaps in data science field and potential opportunities for further research. And being part of the RLD's global, I wanted to showcase, I just wanted to let people know how R programming is used in Botswana because it's a lesser known country to be using R programming. The study followed the PRISMA methodology, which is short for Preferred Reporting Items for Systematic Reviews and Meta-Analyses. For reporting in a systematic review, PRISMA was chosen because it's the recognized standard for reporting evidence in systematic reviews, and meta-analysis and standards are endorsed by the organization. We're going through all the journals and databases such as IEEE Explore, ACM, and Web of Science. Over 2,300 articles were gone through, and they were filtered by the studies that were conducted in Botswana. And after going through the identification phase, it was filtered again to look at the studies that were in Botswana that were particularly using programming. Continue in the screening phase, it was continually filtered to exclude some of the research that only had one research, to be specific. It was continually filtered to exclude the studies that were done before 2010 or 2015 until we got about 18 studies that we concluded having been used. So altogether we saw healthcare, physical geography, conservation, and climatology were the research areas that we used R programming the most, with healthcare being the most, followed by physical geography, conservation, and climatology. The healthcare in Botswana has had numerous research conducted in Botswana in order to improve the efficiency of health facilities by addressing potential barriers. This is part of the National Development Plan, Vision 2036, which aims to move Botswana from a middle-income country into a high-income country by 2036. So the descriptive analysis from data collected from regions of Botswana has helped to understand the actuality of treatment distribution, ongoing treatment research, and health institution management. So looking at the R packages that were used for the healthcare research in Botswana, the forecast package implements automatic forecasting using exponential smoothing, the ARIMA models, Theta method, cubic splines, as well as other common forecasting methods. There's a study produced by colleagues from my university, which adopted a machine learning-based time series model to forecast confirmed COVID-19 cases in Botswana in a 60-day period. To successfully implement the ETS model, the forecast package was executed in R. The OCE package for oceanographic analysis handles the details of discipline-specific file formats, calculations, and plots. It's easy to document work using OCE because its functions automatically update processes, logs, and stores in data, and they store the data in data objects. There was another study which coordinated the Administrative Center of Health District before applying the Vicentries Etholosodal formula for geodesic distance as implemented by the Oceanic package. ggplot is a framework that is another package that allows making both graphic plots and annotations. This package enables developers to build almost any type of plot, from dentograms, network graphs, and histograms. So a research group from the university characterized genetic variations to assess population substructure within a cohort of HIV-positive children from Botswana that is regionally underrepresented in genomic databases. They used annotations in the analysis that were visualized using ggplot2. Another package was the SNP relay package in genomic research for packages used for principal component analysis and identify by descent measures. For the same research that used ggplot, they needed to assess substructure with the Botswana cohort and follow the QC pipeline in the PCA analysis, the principal component analysis, and the independent autosomal markers proved by R-squared coefficiency in windows of 1,000 base pairs advanced 100 SNPs at a time where the time were used for the analysis of the SNP package. Other packages included the IncTools package for containing incidences from biomarker data in cross-sectional surveys. Another one was the analysis of phylogenetics and evolution, which was APE for short. So phylogenetics is the study of reconstructing the past evolutionary history of species, which provides utility functions for reading and writing data, manipulating phylogenetic trees, as well as several advanced methods and manipulating phylogenetic evolution analysis. The iGraph, which is also another package which provides tools for researchers in network science. It contains routines for creating, manipulating, and visualizing networks, calculating various structural properties, importing from and exporting to various file formats. So another study by Novistics and others used it across these, used the iGraph package for visualization. The physical geography research area that also used R programming had some various packages. So the physical geography makes up the landscape, wildlife, and Botswana, and in Botswana that has generated a lot of research for sustainability, such as the Okavango Research Initiative. The research undertakes engaged research and provides training and service in wetland and dryland social and ecological systems with support from the South African Science Service Center for Climate Change Adaptive Land Management. So their packages included the MetLab, the MetAb-R for analysis of metabolomics data, which is the study of small molecules generated by the GCMS, a chemistry analysis operation. Another included the MNLE package developed for fitting and comparing Gaussian linear and nonlinear mixed effects models. A study by Fox classified multi-decadal changes in land cover in the semi-arid Chobe Desert in northern Botswana. And what he did was to calculate the regressions to the datasets. In the package, they used 5 autocorrelation structures: exponential, Gaussian, spherical, linear, and rational quadratic. The 2 machine learning packages, which are the RPART packages, were used for calculating classification accuracy, as well as mapping using the conservation in Botswana aims to evaluate the preservation practices for wildlife and landscape in Botswana, the transition of savanna ecosystems to open shrubland across Botswana, and in particular the western part of the Kalahari presents a considerable threat to the conservation of the important ranching industry. So in order to develop adaptive management strategies, the underlying environment drivers of woody vegetation species need to be better understood. So drivers of the woody vegetation species need to be better understood by driving the responsible diversity and abundance of woody vegetation. So clusters, the RAFLID, R-pod package, and partikit were all used to create dendrograms and also for machine learning classification. And some studies even use them for cluster analysis. The climatology research in Botswana includes measuring the change in rainfall and rainfall composition over a period of time. Historically, Botswana has depended on rainfall for crop production, livestock, as well as water source and keeping the air moist because it's a desert country. So most of the research in climatology didn't use R packages, but instead used base R packages, which are the statistics and mathematical operations. Some used R operations for rescaling, which is the rescaling range analysis for residual series obtained after filtering periodicities. So the base R was used to assess net atmospheric methane fluxes and controlling environmental factors in the Okavango Delta. The precipitation concentration index, the Mann-Kendall trend, the tail-sin slope estimator, autocorrelation function, and relative percentage change methods were adopted for data analysis, were performed using base R. Others were descriptive statistics, trend analysis, and change point detection techniques were developed using base R. So the techniques, as we can see, research in Botswana that have used R programming have used it for statistical, descriptive analysis, and exploratory analysis to demonstrate the patterns in weather and national program evaluations and change in the landscape. So the variety of data visualization, mapping, biomarkering, and genomic operations used explain the many R packages used in the healthcare research. The reason for R packages used in certain research areas may be due to certain R packages being made for operations, for example, the SNP Relate for genomics and AID package for the phylogenetics in healthcare. So looking at the graph, the most used R packages across the different research areas is the R-pod package, which we can see in purple, which may be due to the methods and operations for classification modeling. So classification showed to be the most used analysis in the research in CAROT, R-pod forecast, and partikit. The partikit R packages used for classification, regression, and Gaussian modeling. This proves that R programming can be used for machine learning, contrary to the belief that machine learning isn't appropriate for R, but instead used for R programming. No, used for Python programming, sorry. So looking at it, we can see as R is an open source software. It's open to developers to create packages to have operations to perform certain functions. All the R packages that have been used in research have been created by developers outside Africa. To the best of my knowledge and what I've seen, there are no documented R packages developed by people in Botswana or in Africa, if they are there at all. However, the R packages used are necessary for carrying out particular operations such as classification, regression, and MetaB R. So the infrequent use of R programming research in Botswana may be due to the limited R experts in R programming or who are not proficient in it. And a reason for the limited R proficiency may be that in academia, SPSS, Stata, and Atlas TI are more common in data science. So in Botswana, very few people know about R programming for data science and for using it in their project work. And they still consider it as a statistical tool despite that it's being used for data science. So as far as the future work, R research, which uses machine learning, can be evaluated The study also didn't look at the statistical models used by each study due to the number of models per study. And this can be used as a future work to consider which statistical models are more appropriate for the research area. Thank you so much for into questions.
Thank you, Simisana. While the judges just finish their collaboration, I have a question for you actually, which is about how much R is taught across the universities in Botswana. Do you have any idea of how many people would know it if they're coming into this sort of field?
Not the people who would only know about it will be students and statisticians. Like at my University of Botswana, only the students who are in population studies would know about it. And where I'm studying right now, which is across the country, it's mainly under social science as well. So not a lot of people know of it. They only know of Python programming.
So Python's quite widespread across research in Botswana, right?
Yeah, it is, in most of Africa.
Great. Do we have any other questions for Simisana?
I have a question. Probably I
sure.
missed this, but when you talk about research in Botswana and looking at the papers, how do you determine which papers from Botswana? Is it the people with the final authors or the first authors affiliation? Or how do you categorize whether the research is from the country?
The study had to be based in Botswana. All the
Oh, right,
research
right.
area, everything at the data and what they were looking for had to be done in Botswana.
Any other questions?
Yeah, I have a question. I'm not sure if I missed this at the start, but how do the research areas that R is used for, or the packages that are used compare to other countries? Do we have any information like that available to compare?
The best way to compare them would be using the CRAN. The CRAN is available online, and the CRAN does state who the developers are, the maintainers are, but they don't really say where they develop from, like which countries it's developed from. But they are available online.
Thank you.
Okay, we better finish up there before we run out of time for the session. So let's thank all three speakers again for their talks. I've really enjoyed hearing about a very broad range of research topics from our ECRs, and it's been great to be part of that. So our judges have been conferring in the background as the session's been going on, and they have come to a decision about the winner. And the winner for this session for the best presentation is Felicia Bongiovanni from WeHi. So congratulations to Felicia. And thank you again as well.
Thank you. Oh, sorry, I didn't know I said something.
You can say something.
I just wanted to very quickly say thank you. I didn't expect that there would be an outcome so quickly.
We're efficient over here. And well done again to everyone, and thank you for coming along. And thank you once again to our judges, Julie Cook, was my names, Lauren Smith and Mahsa Salehi, for helping participate in this session and for judging all of our speakers. And with that, I'll close off this session and I will hand over to the next chair who's already here. So thank you a lot. Thanks, everyone.